and I look forward to your talk.
Hi, thank you very much for the introduction. I don't need to say much about myself then, which is great. So as you said, I'm going to be talking about this DNA extraction method suitable for shotgun metagenomic sequencing. And this is based on a recent publication of mine. So if you want more information and to access the paper in your own time, I've also provided a QR code for you. But today we'll be going over the background and aims of this research, methods and results, and obviously the conclusions and some future work. Okay, so I'm just going to give a brief background on the skin microbiome and its importance in skin health. So the human skin microbiome is a complex ecosystem, and it's organised into microbial communities at different body sites. So these communities include bacteria, archaea, viruses, and fungi, and they participate in the host's skin physiological functions and immunity, and are therefore really important for our skin health. However, early life perturbations in these communities can cause skin dysbiosis, and this can impact our skin health. Several dysbiosis-related conditions of the skin include atopic dermatitis, eczema, and psoriasis. So my research focuses on the infant skin microbiome. So the main point that I want to make here with this slide is that the skin microbiome, especially the infant skin microbiome, is still relatively understudied. The bacteriome is the most studied community within the microbiome, followed by the microbiome, and then the virome and the archaeome are still very understudied, and a lot more work is needed. So there are just some examples here for you that I've just left here for you to briefly read through, and it's just examples of the microbiome communities in infants, and also some early life factors and longer term factors that can impact our skin health. I'm not going to go through these today as I don't have enough time, but if you would like more information on this and the infant skin microbiome in general, I've put another QR code here which should take you to a recent review paper of mine as well. Okay, so how do we actually study the skin microbiome? So as was just mentioned, 16S RNA gene amplicon sequencing is mostly used to classify the skin's communities. However, shotgun metagenomic sequencing is emerging as a preferable method now. So why is this? So 16S has lower taxonomic resolution, and it classifies microbiota to genus level, but can also go up to species level. It only classifies bacteria and archaea as they contain the 16S target region. It also doesn't really provide functional information on the microbiota. So in comparison, shotgun metagenomic sequencing has a higher taxonomic resolution as it can classify microbiota up to strain level. It classifies all microbes present within the skin, and it can provide functional information as well. So this diagram is just a nice way of visually showing this. So you've got your mixed microbial community, you have your extracted DNA, and at the top box here is amplicon sequencing, and you can see in the colors these targeted regions in the genomes, whereas metagenomic sequencing, you can see the whole genome is sequenced. But there are research challenges with skin. So skin has low microbial biomass. There's also lack of skin optimized, commercialized DNA extraction protocols available on the market. So this challenges the extraction of sufficient quantities of DNA for shotgun metagenomic sequencing, especially those that are of higher molecular weight as well. So this is particularly true for longer read technologies where more input material is needed, and that is also of high molecular weight. So we aimed to address this need by optimizing a high-throughput automated DNA extraction method for recovery of high molecular weight microbial DNA from skin swabs. And this was validated using swabs from adult volunteers and also babies that are enrolled in the pregnancy and early life study. Okay, so I'm just gonna go through very briefly a series of experiments that I performed before finalizing a method. So to determine an effective initial diluent in recovering extracted bacterial cells from swabs, that was spiked with plate colonies of gram-positive and negative bacteria. So I was using PBS and water because this is what's mostly used in literature. So I thought I'll do a comparison to see what has the best results. I measured the DNA quantity using the qubit, but overall there was no significant difference in the extracted DNA quantity between the diluents. I then compared the bacterial viability in these diluents, and this was because I wanted culturing to be performed effectively alongside the extraction method. So the same gram-negative and positive bacteria were grown, but in the form of liquid cultures. These were resuspended in PBS or water, and then serial diluted and plated, and the CFU per mil measured. So again, there was no significant difference in the cell viability between the diluents. So going forward, I decided to use PBS as this was the most widely used diluent in literature. So using PBS, I then compared different extraction lysis methods to identify one of the methods that would result in the highest DNA yield and highest molecular weight from both gram-positive and negative bacteria. So the finalized method included an overnight lysis with a lysozyme, a further heated off-board lysis step, and a bead beating step using a tissue lyser. So the molecular weights ranged from about 22 to 31,000 base pairs, and the DNA yields range from 37 to 104 nanograms per microliter. So I then performed this finalized method on 24 adult volunteer swabs and 10 pulse study skin swabs, and also the cultures from these swabs. And I sequenced them all using shotgun metagenomic sequencing on Illumina and Oxford Nanopore platforms because I wanted short and long reads. So this was all to verify successful harvesting of DNA and taxonomic profiling with real swabs. And this is going to be the focus of today in terms of results. But if you want more information on these previous experiments, you can just follow the QR code, which should take you to the publication. Okay, so in terms of the swab DNA yield, DNA concentrations from the adults and babies ranged from less than 0.5 to 10.5 nanograms per microliter. So the DNA was successfully extracted from all the baby samples, but only 15 out of 24 adult volunteer samples had DNA extracted from. So just to note, these swabs had all been incubated for 18 hours during the overnight lysis. In terms of the culture's DNA yield, the swab cultures recovered bacteria from all the adult skin swabs and 4 out of 10 baby swabs. And concentrations from these averaged at 79 nanograms per microliter. So I mentioned just now that some adult swabs didn't yield DNA. So these were the lower end here that I just previously showed you. And they were all incubated up to for 18 hours. So we decided to compare DNA yield from extracted swabs after different overnight lysis incubation times. So the samples were randomly incubated for either 18 hours, 20 hours, or 22 hours. And what you can see here is that there was a significant difference between 18 hours and 20 hours and 18 hours and 22 hours in terms of the DNA yielded. And you can see as well from 20 hours onwards, the DNA concentrations yielded are then very consistent. So future samples should be incubated from 20 to 22 hours to obtain this higher DNA yield. Okay, so in terms of the actual sequencing data, this is the pipeline that I used to analyze the data. Again, I don't really have time to go through this in depth, but to summarize, the data was cleaned and run through Kraken 2 and Bracken for taxonomic profiling, which is a read mapping method. But overall, both the Illumina and Nanopore data generated enough reads, allowing downstream taxonomic analysis at the species level. So I'm just going to go through the baby data today. So these are taxonomic profiles of the skin swabs, which are shown in figure A, and then the cultures of the swabs, which are shown in figure B. So these are from the 10 babies, and it's also showing the Illumina and the Nanopore data. I know this is a little bit hard to read on my screen for you, so just kind of the main points to take home really is that the baby swabs only displayed bacterial diversity, whereas the adult swabs had bacterial diversity, viruses and phages present as well. Baby skin swabs contained more Streptococcus and fewer Staphylococcus species when compared to the adult swabs. Babies also presented with Bifidobacterium longum, breve, and bifidum. So these aren't usually found on the skin, so they're not typical residents, but they are common residents of the gut and breast milk. And so this likely demonstrates transient skin contamination on the babies. So this is nicely demonstrating those early life factors that I briefly mentioned earlier on coming into play. So analysis of the taxonomic profile from the cultured samples exhibited less microbial diversity than the skin swabs, which is expected, but they did also confirm presence of species that were identified in the swabs too. Overall, the Illumina and Nanopore platforms did identify very similar microbiota profiles within the swabs and the cultures. So I then wanted to do a platform comparability. So this is an alluvial plot showing the percentage total abundant species counts between Illumina and Nanopore data for the skin swabs and cultures. So the y-axis is showing the percentage there of the counts, and then this first bar is the Illumina culture data, the second bar is the Illumina swab data, the third is the Nanopore culture data, and the fourth is the Nanopore swab data. And the adjoining lines between them all are the common species throughout each of these 4 data sets. So overall, Illumina and the Nanopore platforms did identify similar profiles for both skin swabs and cultures. They also had comparable counts for the most abundant species. The swabs did show more diversity than the cultures. So, for example, you can see here, this is the swab data set versus a culture data set. You can see there's a lot more diversity. And both platforms' performance were comparable. So finally, I also wanted to identify a suitable sequence depth for species and MAGS recovery from skin. So I sequenced 10 baby samples at 1 gigabase pairs and 5 gigabase pairs of data. So if we just look at figure A here now, so sub-sampled 5 gigabase pairs of data reads, I sub-sampled 5 gigabase pairs of data reads to compare the species recovery at the different depths. So there was a significant difference in species recovery between 100% reads used and 40% reads used. So 100% reads used represents 5 gig data, and 40% reads used represents 1 gig of data. But there wasn't a significant difference in species recovery between 5 gig here and 2.5 gig, which is about 50% reads used. I then identified MAGs, which is in figure B here. So more MAGs were recovered at 5 gigabase pairs, but there was no significant difference in the MAGs recovery between 1 gig and 5 gig. So overall, 5 gig was adequate for phylogenetic analysis of the skin and also to generate MAGs, but a higher sequencing depth would be better suited for MAGs generation. Okay, so to conclude, an optimized extraction, shotgun, and analysis approach can effectively characterize the skin microbiota from both adults and babies. And this method can be applied for a more in-depth analysis of cohort studies, and this can allow identification of their taxonomic and functional changes of mothers and their babies over time, which should allow comparison to other body sites as well, such as the gut. Robust microbiota profiling, especially in less well-studied niches like the skin, is important for the development of new methods to alter the microbiome composition for skin health. So this is tying into my future work now. So I'm currently applying this method to a larger longitudinal set of the Pearl study samples, and I'm looking at infants from birth up until 2 years of age. And I'm comparing microbiome evolution. So I'm looking at the developing infant skin microbiome to the mother's vaginal microbiome to try and look at this evolution occurring. And I also want to assess changes to the infant's gut microbiome and how this might impact the microbiome development and skin health. And hopefully this will also enable us to determine nutritional interventions that could help treat any skin dysbiosis occurring. And also, from this, I'm wanting to isolate and characterize bacterial isolates of interest so that we can look at these functional traits and importance in the skin. And finally, which is probably further down the line, try and predict how we can alter a microbiome to actually maintain skin health throughout the life course. Thank you very much for listening. Just want to thank both my groups, the Weber group and the Hall Lab group. Thank you very much, everyone.